In this video, we're going to look at antibodies in the immune system. First, to start off, we have two types of immunity to fight against pathogens. We have cellular immunity, which will fight most pathogens, will eliminate virally infected cells, and it's mediated by T lymphocytes. And we have humoral immunity, which is effective against bacteria, and also the extracellular phases of viral infections. These are regulated by our antibodies, also known as immunoglobulins, and these are made from beta lymphocytes. Now let's just look at the humoral immune system in general. And so the immune system is triggered by what we call an antigen. This is a foreign macromolecule, a protein or a carbohydrate generally. Now on beta cells or B cells, we have immunoglobulins or antibodies sticking from the surface. Now when a beta cell encounters an antigen, and if the antigen and immunoglobulin are compatible, you will have a binding event. Then the beta cell will engulf this antigen antibody complex and it's become activated. And T cells stimulate the B cells to become activated. And B cells will do two things. One, you'll create other B cells that will make antibodies that will bind to this antigen. And you'll also create memory B cells, which will recognize this antigen for several months or years down the road. So if you're infected by the same antigen, then you can have a much faster response time to that foreign macromolecule. And we can see that here in this graph. So we have antibody concentration on the Y and days of infection on the X. So if we add antibody A, we can see that after a week our antibody concentration is at its maximum, right? And after this time we have made memory B cells. So if we're infected by antibody A again and a new antigen we're going to call B, you can see that after a week the amount of antibodies to A is roughly 10,000 times greater than the antibodies we have to B. So since we have these memory cells around, our response time to that antigen is much faster and much more complete than to a brand new antigen. Now let's look at the antibodies or immunoglobulins. And all antibodies have the same general structure. They have 4 subunits. 2 we call light chained because they're lighter than our 2 identical heavy chains. And the different subunits all associate via disulfide bonds and non-covalent interactions. So these 4 different subunits are not covalently connected. And we have discovered that there are 5 different classes of immunoglobulins, and the difference is in the heavy chain. So IgMs are your Y-shaped molecules, and they're centered around the J subunit. You have 5 of those. IgAs can be monomers, dimers, trimers, or tetramers, and they're really good against microorganisms. And they're generally the first antibody to be secreted in response to an antigen, and you'll find these in your intestine. IgGs are your most common. They're both in your blood and extravascular fluids. IgE is in blood in minute concentrations, and these are the protections against your parasites. And it's also thought that these are the antibodies that cause your allergic reactions. And you also have IgDs. They're small amount, but we don't really know what they do yet. There's no clear function for them. Now we're going to focus on IgGs because they are the most prevalent. And so here's a picture of the IgG. And you have 2 different segments of your antibody. You have the Fab and you have the FC. Now you can separate the FAB and FC regions by using a molecule called papin. Papin is proteolysis, so you're cutting your peptide bond, and where it cuts is right here, and that's where we say we have a Fab and an FC region. So the FAB region is called AB for antigen binding region. You have your entire light chain, which is shown here, and you also have the N terminus of your heavy chain. So at the end, this red is where you're binding antigens. The FC, the C is termed because this part of the protein forms protein crystals super easily. This contains the C terminus half of your heavy chains. And these FAB and FAC region is connected through this hinge. And now you can also see here we have our disulfide bonds that are holding all these subunits together. And so the antibody has two various regions. We have the variable region up here, and we have the constant region. And only one beta cell produces one type of antibody. But your body has multiple different types of B cells, so that's why it's polyclonal. We have a lot of different antibodies in our body, but only one B cell will produce one type of antibody. That is, it will recognize one type of antigen. Now let's look at the secondary structure of our antibodies or immunoglobulins. And immunoglobulins have what's called the immunoglobulin fold. You have 3 to 4 anti-parallel beta sandwich here, which is held together by disulfide bonds. So the yellow here is your disulfide bonds. And variation in our antibodies is seen at these hypervariable loops, which is green here. So these loops, the differences in these loops allow antibodies to bind to different antigens. And again, these are in the FAB, or the antigen binding site at the tip of the FAB. Now, when an antibody binds an antigen, this is all non-covalent interactions. So this is all due to hydrophobic interactions, hydrogen bonding interactions, and ionic interactions. And once bound, it's very hard to get an antibody off because they have extremely strong binding. Their dissociation constants are 10 to the minus 4 and 10 to the minus 10 molar. Now, antibodies, they're called divalent. That means they can bind two things at once because they have two FAB regions in that shape of the Y. So what actually can happen is that if you have antibodies and antigens, you can form like this network where an antibody is binding two antigens at once. Maybe another antibody binds another type of antigen on the same site, or a different site rather, and you just have this network of antigens in a cross-link lattice. And this is thought to help speed up the body's response to antigens, having this type of cross-link lattice. Now, when you are exposed to a new antigen, your body won't create a new antibody. That's kind of weird to think about, but all the antibodies you need to look at every type of foreign antigen, your body already has, right? So our body, on estimate, can create 10 to the power of 18 different antibodies. However, each individual will only make a small fraction of that in their lifetime. And this massive diversity in antibodies is due to how beta lymphocytes develop. They have a lot of genetic change that happens there. So for antibodies, each chain in our antibody is encoded by a lot of different DNA fragments. And these fragments are joined together after the fact by somatic recombination. So before you even transcribe or translate, your DNA to make your antibodies is already being scrambled and connected in different ways. That's how you're getting some of these variable regions. And so you have a large number of possibilities there due to the different ways you can connect these segments. But also, when your beta cells divide, the rate of gene mutation inside these cells is greatly increased. Every generation, you have on average one amino acid that changes. We call this somatic hypermutation. So that's how your variable regions are so different from beta cell to beta cell. The machinery that is normally there to protect against DNA mutation might not work correctly in beta cells. So you have different amino acids on your Fab regions, and this allows for different antigens to be recognized. Now, antibodies can only bind to foreign material in general, but it is possible for antibodies to bind to macromolecules that are part of yourself. We call this autoimmune diseases. And so first, let's just realize how amazing it is our antibodies normally don't attack ourselves, because we have tens of thousands of different macromolecules, yet our antibodies know that those are part of us and not foreign. This can be a problem, though, if you get, let's say, a tissue transplant. Now your body will bind to this new tissue because it will recognize it as foreign and start to attack this tissue. And so it's not well understood why our antibodies don't attack us. But the idea is if beta cells or T cells recognize molecules as part of us, they are destroyed before they can fully mature and create memory B cells and create these antibodies to attack ourselves. And this process happens from birth and onwards. And the ability or the loss of this ability will lead to these autoimmune diseases where your body will attack yourself. Now, some tissues are generally targeted more than others, and autoimmune diseases often happen due to an injury or disease. So let's say you had a head injury and the blood barrier to your brain is compromised. Well, antibodies could get into your brain and they would recognize antigens they've never seen before. They would recognize these as foreign, and then your body would attack your brain. That's just one type of example. But here are some other diseases. So in your adrenal cortex, your thyroid gland, pancreatic beta cells, connective tissue, DNA, all being attacked by your own antibodies. And that's it for this video. I will see you in the next video.